phases. One in the batch scale and second in the reactor scale. So we picked up this problem just because there is a scarcity of water in and around Varanasi district. A lot of the places are found heavily contaminated with different kinds of metals, metalloids, and pollutants. Especially the belt of Bihar, West Bengal, Bangladesh, Pakistan belt, Balia belt, they all are heavily contaminated. So we decided to work on a eco-friendly technology rather than a synthetic technology. That too, which can work upon the large scale or the industrial scale. So the topic of bioremediation of wastewater by macroalgal system. Here's the introduction part of it. There is a surge in the industrialization, urbanization, and globalization in the last 20 or 30 years, last 2-3 decades, and these surges actually have contributed significantly in the emission of greenhouse gases and then waste generation.
Sorry to interrupt. So can you make the slide full screen?
Oh, sure.
Thank you.
Now is it fine? I have made it full screen. Is it okay now? Can you hear me?
Sir, actually, we would like to have the slideshow. So can you do the slideshow in
I
full
have
screen?
I have already done it in the slideshow full screen.
So it's not showing in the video. Can you do it again, please?
1 second. Now I'm on slide number 3, which shows greenhouse emissions and wastewater generation.
Yes, sir. Yes, sir. It is showing that, but we want it full screen. So can you go
I
to
have
the
already
slideshow tab and
made it slideshow type. In my laptop, I have
okay,
already done it in the slideshow type.
actually, it's not yet full screen actually. So there
Again, I have
must be some technical glitch.
again I'm doing that. I'm just escaped from the same menu, and now I am
Yes,
now
sure, sure.
now it started working.
No, sir. No, sir. You can continue as you are doing. No problem.
But in my laptop, it is showing like uh, completely in the slideshow mode, and there's animation in the every slide. So they're also moving. So now,
Okay, but it's not showing in the video, but you can continue, sir.
oh, that's fine. Industrialization, urbanization, globalization have made a surge in the last 2-3 decades. That has led to the serious enhancement in the emission of greenhouse gases and the volumes of wastewater. So now the next slide, what are the activities which are actually involved in polluting the things? So it is as simple as the urbanization has led to the production of several anthropogenic actions which produce huge amount of wastewater. So I have tried to show on the y-axis that how this volumes of wastewater is getting increased in every 10 years or in 20 years.
Sorry, sir. Sorry to interrupt, sir. The slides are not moving. You are still in slide 3. So I guess there is some network issue.
So what can I be done now? Now I have removed
Yeah, now
my.
it's fine. Now it's fine. Now it's
Now
fine, sir. Yeah.
I have also removed my presentation mode. I'm back to
Okay,
the
okay.
same
You
common
can
mode.
present like this, sir. Thank you, sir.
Oh, okay. So what can I do? I can magnify the font from here. That may suffice the purpose. 1 second. So now there is a surge in the production of wastewater all across the globe, and the values are highly tremendous in volume. So now 80% of the wastewater is released into the water bodies without proper treatment. It is because most of the treatment technologies require huge amount of input cost, and majority of the industries are not ready to bear it. The next one is excess discharge of wastewater increases the nutrient levels, causing the eutrophication. Eutrophication leads to the threat to fishing and real estate activities. Approximately, there's a data that 2 billion US dollars usually loss occurs due to eutrophication. Now, this leads to lower economy. That in the lower economic countries, they only treat about 8% of the total wastewater. Middle economic countries, they treat 35% of the wastewater, and the higher economic countries, which are very small in number, they treat 75% of their wastewater. That has led to a scarcity of the natural pure water. Moreover, this also discards the recycling of the wastewater once one or more than one cycle, and that leads to the tremendous loss of the water directly into the natural water resources, that makes it contaminated. So this is the CO2 emission in terms of metric gigatons in India, US, and China. I've just compiled all the type of the countries, tried to show all the countries, and there is an increase in the global temperature above of around 0.8 to 1.2 degrees centigrade above the set level for industries. During the Paris Agreement in 2015, it was decided actually to reduce the emission of greenhouse gases, but till date, no such strong action has been taken up. And the limit of temperature increases below 1.5 degree centigrade. Now, this all leads to cumulative effect goes to the global house warming, and there is a water level rise in of the oceans. That water cannot be used for any purpose, neither for the drinking, nor for bathing, nor for making any construction activity. But since the water is in the large part of the land, it is also capturing the huge amount of the land, and it is keep on capturing the new fractions of the land. So that actually leads to the land loss as well. There is a huge pollution, and this is leading to the high increased temperature gradient all across the globe. So as a matter of fact, we decided to use such a technology which is completely eco-friendly in nature with the two notions. First, we need, the primary objective is to treat the wastewater. Now there are the different categories of wastewater. There is a domestic wastewater, industrial wastewater, municipal wastewater, sewage, and then industrial, then effluents. Effluents categorize, the categories of effluents are different. You may have the effluent from the dairy industry, a smelting plant, metallurgical units, pharmaceutical plants. So a versatile solution of eco-friendly type is required. So we decided to work upon the macroalgae this way. It is because they can be grown easily in any type of the wastewater. The input cost is too small, and the reactor which we fabricated on a lab scale was very much inexpensive in nature. Slowly I will show all the slides what we have done. Some computational analysis we also made just to elucidate or just to find out that how we can use the data which has been published in different research papers to make some good pathways which a researcher can follow to obtain the maximum yield. So the two objectives were first to treat the wastewater and to produce the biomass in bulk using the same wastewater as media. So what actually happens when we increase the volume of the media and what we obtain as the biomass as a byproduct, that can also be sold in the market. So two objectives we are getting. We are treating the wastewater by the eco-friendly route, most probably without the intervention of any funding into it, and the biomass which we are getting after the treatment will be so enriched that it can be sold at a huge cost in the market, in the open market, as a source of protein, as a source of bioorganic fertilizers. So slowly we started on that. So what was actually why we tested and decided to work upon the macroalgae? It treats the wastewater simultaneously recovery of nutrients in the form of biomass, high nutrient uptake rate along with high CO2 fixing potential, high photon to biomass conversion efficiency. More than 3% we obtained in the photobioreactor which we made in our lab system. And it was only having the two assemblies. One, we use a pump, submersible pump, to propagate the oxygen inside the flask or inside the small reactor, and some few tube lights of different wavelengths. Now conversion of biomass to biofuel can be done. Some other advantages of that, or bioethanol or other energy sources, conversion of biomass to biofertilizer, electricity generation using the biomass. This actually we call as microbial fuel cells. The third point, we call it as microbial fuel cells here. And here we are not only the macroalgae we have used, we have used also different kinds of bacterial system. And this problem is more relevant to the liquid waste and to the solid waste. Both we have handled in the microbial fuel cells for the production of electricity. So this is the type of figure which we used. This data has been already published in Biochemical Engineering Journal. And what we have actually tested here, we just made a kind of reactor system, and that reactor system was photobioreactor PBR type, very easy in the assembly, and there was nothing complex there. And we just added some nutrients or utilize some kind of wastewater, added the macroalgae there. There was a cultivation phenomenon which actually happened for a few days. Then we have a recovery pond system kind of assembly where we recovered all the biomass, and biomass was concentrated by using different downstream processing, then drying, and the algae biomass was separated out. While the algae grown in that submedia, that wastewater, and there was a huge pollutants, huge amount of pollutants or huge content of pollutants which were actually removed, completely removed by the growth of macroalgae. This was one idea to work upon. But since the experiments involve a huge amount of cost, they involve a huge amount of electricity, continuous monitoring, as well. You also need a huge amount of reagents to cater the needs of macroalgae during the growth period. Say, for example, I need to maintain the pH, so I need some acid and bases. I need some sort of nutrients if there is acclimatization phase to boost the algae growth. So what should be the way? It involves a huge amount of cost. On an average, experiment costs around 8 to 9,000 rupees at the lab scale. So if I'm going to implement this technology or in a river or in a kind of pond or lake, it may cost around lakhs or in crores. Government was not ready to defend the case. So we decided, why not to work upon a technology which can guide us properly. Before doing the experiments, if we get the idea what experiment should be done, suppose I need to conduct 1000 experiments to find out which is the best pathway for me to work upon and to get the maximum yield of biomass along with the maximum purification of the wastewater. What should be done? Either I carry out the experiments 1000 times with different optimization conditions with different parameters, or just to study the research papers and to find out the different kinds of variables and to identify the trend which is available in their experimental readings. So that is why we actually used the different kinds of machine learning technology in the growth of microalgae system. Here are these. The machine learning algorithm can quickly analyze the extensive set of data. We utilize this actually technology. This is it's a simple algorithm. There are different patterns of these algorithms which have been used by several researchers across globe. So some references I have tried to cite. So it is all about the application of machine learning in the field of macroalgae. In the last 10 years, what we identified in the literature review, that several people have worked on this idea. This idea is so unique that without doing a large number of experiments, say 1000, 2000 experiments, by only conducting 10 or 20 experiments, you can get the pathway of the best pathway to yield a high amount of biomass, to achieve the highest amount of biomass, along with the best possible recovery of all the nutrients and the purification of water. So how the people have done, so just some themes I have tried to summarize here. Predicting the critical factors for macroalgae biomass and lipid production. This has been done by one scientist in the year 2021. Then modeling of the chlorella species harvesting, so on and so forth. Several others have done upon the growth of the macroalgae in the raceway pond system and the microbial consortia. What is their actually theme? Why they worked upon? But the research, and what is the lacuna here? The main lacuna was the research related to the application of ML for optimizing the microalgae based wastewater treatment is completely missing. They have only tried to optimize the parameters, but they have not studied it experimentally. One more thing, it can determine the best combinations of predictor input variables for increasing the biomass productivity and wastewater treatment capacity of the macroalgae. Suppose macroalgae is only capable to treat 10% of the wastewater volume by volume or percentage by percentage. Now, by giving the best combination of the variables, which we call in the mathematical language as predictors, it may treat 20% of the total wastewater or 25. Same algae, no chemical reaction is required, no mutation is required. Only by fluctuating the experimental conditions, we have done the work. It can even generate new information from already reported data, which is expected for new experiments. So the already reported data helps us or guides us for performing the new experiments in the right way. So this actually saves our time and money. Now, patterns detected from these algorithms can assist in constructing high throughput experimental designs. The designing of the experiment is a very critical part of research. If my design is right, in very less number of experiments, I will get the right answer. If my design is wrong, then I may not get the appropriate answer. And so what was the methodology for that? So we just filtered several thousands of the manuscripts altogether. And we got some very good papers out of this. One of the manuscripts was published in the journal like Water Research. It has impact of 12 and almost 500 papers we tried to cite in that manuscript. So what we did actually, how we started preparing the complete data set for analyzing by using machine learning. In machine learning, we have also tested different kinds of algorithm. So the approximity or the accuracy of entire model depends on also upon the type of the algorithm which we have selected to treat the problem. So what is that? Experimental data set referring to nitrogen removal efficiency, phosphate removal efficiency, and biomass production were prepared initially. So we did this critical study by summarizing several research papers. Data set was prepared by collecting the data point from previously published research articles containing around 500 data points. So we just selected the time zone of 2010 to 2022. The input or predictor variable considered for evaluation and their range or identities had been mentioned in the next table. Now the response variables like NRE, that is the nitrogen removal efficiency, phosphate removal efficiency, and biomass production, we are grouped into two categories. High level, that means the higher maxima or the maximum of the equation, and the low level means the minimum of the equation, taking the median as the threshold value. Now the values above the threshold we are grouped into high level class and low level class. And this how we just fabricated the entire experimental design. This is how. So we selected the predictor variables, that is the influencing variables which are influencing the total data. This we have taken from a journal. We have also tried to cite the source. Somewhat out of font it has gone, but we have tried to cite it. So now the macroalgae class where have been shown here. Then wastewater sources: domestic, municipal, agriculture, industrial, synthetic, and livestock. Cultivation type was autotrophic, heterotrophic, and mixotrophic. And then culture type was macroalgae or macroalgae bacteria consortia. They both were taken together just to check the efficiency of the model and how much we can obtain more the surplus ahead of the macroalgae. Then pre-treatment method is primary, aerobic, anaerobic, and autoclave type. That means these primary treatments we have done, pre-treatments. And then it's 4 types we have done. Since autoclave is more or less more energy expensive, so we tried in most of the experiments we found that it is more effective in getting the highest yield and in treating the wastewater. Then the reactor type is actually flask or PBR. Then the CO2 content we try to maintain in a zone of 0.03 to 45%, temperature 9.3 to 35. Then these are the predictor variables: cultivation period, initial evoluculum level, pH, light intensity, photo period, and then reactor volume. For maintaining the photo period in our synthetic simulated reactor system, we use some timer circuits to control the photo period, to control the time zone. The reactor volume was around 100 to 50,000 liters. It just scaled up using some equation, and then initial phosphate content, initial nitrogen content, so on and so forth. The different contents were also analyzed. So this is the processing. All the missing values were removed. Most of the research papers or a majority group doesn't report all the things, all the values altogether. So missing values we removed from the table while making this, while making the table of these data, these parameters in different research papers. We removed those values which were missing, and only valued values were used for creating the data set. Units of all the predictor and response variables were also standardized. Hot coding technique, it's a very elite class of technique which encodes the categorical variables into the continuous variables. Then the correlation analysis was applied for detecting the significant input variables. That is the Pearson correlation coefficient we used. Here R indicates the Pearson correlation coefficient. X and Y indicators are input and output parameters, and N is the total number of data points. Standard equation we have used. Parameters not strongly related to macroalgae growth were omitted. That means we also studied the conclusion part of the manuscript, and we identified that those parameters which are not directly related to the macroalgae growth, so they were omitted from the final data set, just to ease the problem and to remove the complexity. So that is how we worked here. This is the actual schematic graph, or you can say a PFD, process flow diagram, that how we conducted the experiments. We have the raw data collection point, then the data pre-processing was done. That means minima, maxima, followed by the removal of missing values, followed by the removal of those values which are not directly influencing the growth of the macroalgae. Then we have data splitting or the testing subset. Then we have training subset. That means whatever data we got, we divided into 5 fractions. 1 fraction was spent for the testing of the subset data, and 4 fractions were spent for training of the data. That led to the model construction, model validation, then final validation, and final model was predicted. Accuracy of the model which we obtained was around 80%, sometimes 90% or 84%. That all depends upon the experimental condition and the different values which you are taking in the maxima and minima group, and what you have removed also sometimes impact the accuracy of the model. So now the hyperparameters which were selected were including the split criterion. These are the functions which you use in the MATLAB easily. Minimum leaf size and numerical variables to sample were optimized. Optimization was performed using the optimized hyperparameters function keeping value all. That means all has to be considered. CV partition was used for training and testing the subset. Accuracy of the decision tree models were determined using confusion matrix. That I will show in the further results what we actually got. These are some scientific terms or the more or less technical terms which is actually being used during the coding and to make a decision tree. Influence of the predictor variable were determined using predictor importance and plot partial dependence function. So results and discussion about it, what actually happened. So these are already published data And this is a kind of decision tree which we actually got using the algorithm of machine learning technology. So this decision tree which actually indicates the classification of the biomass production. The blue line actually indicates we read this tree from the top to bottom, not from the bottom to top. It is from top to bottom. So they are the branches which you can see in the form of green lines, red lines, and blue lines. Tree, then nodes, and then sub nodes you can find. I have tried to show those lines which are not actually good in getting the biomass production. So all the red values indicate that, or the red lines indicate that, if we follow this pattern of nitrogen to phosphate ratios followed by the photo period or some other factors, the pathway, that red pathway indicates that you will be achieving a very poor growth of biomass production, or biomass production in that vessel. In case you choose the line or the pathway which is being shown by the green line, that may yield a very high amount of biomass. So without doing experiments, we did this by the help of machine learning and analyzing the data set of the in the given research papers of the last 10 or 12 years, 2010 to 2022. The data was actually published in the Journal of Environmental Chemical Engineering. So what we actually did, I'm at slide number 20. Is it changing in your system?
Yes, sir.
Okay. So the title of the slide is predictor class, high, low, and then low, high actual classes. Fine. So that means it's some kinds of confusion matrix for the biomass production. So just I have tried to, what we tried actually is to classify the data points. So 7, 11, 43, 35, they indicate the data points. Some values came to the lower side of the parameter and some came in the upper zone where is the maximum of the equation. So accordingly, we make a confusion matrix. Final accuracy of the model was around 81.25%. So this is all around. We tried to develop a relative chart or bar graph, that is the relative importance of the predictor variables or input variables on the biomass production. So on the Y, we tried to plot the relative importance, and the input parameters I've tried to show on the x-axis. It is the flask size, sometimes the photobioreactor type, then raceway pond system is there, autotrophic mode, heterotrophic mode, mixotrophic mode, aerobic, anaerobic, autoclaved water, primary treated water, agriculture, domestic, industrial waste. So the different classes we obtained, and the relative importance, we just found that some of the parameters like temperature, inoculum level, pH, light intensity, all on the extreme right hand side of the graph, photo periods, N upon P ratio, they have significant impact on the biomass production rather than the type of the PBR used, mode of the algae, how it is consuming the pollutants out of the wastewater, say autotrophic, heterotrophic. More or less, these experimental parameters like CO2, temperature, initial inoculum level, pH, light intensity, photo period, nitrogen to phosphate ratio were actually involved in the biomass production. So variation of this figure we have tried to show the variation of high and low level microalgae biomass production with the descriptor variables determined via partial dependence plot. That is how the CO2 is affecting, how the temperature is affecting those variables which were actually involved, massively involved, majorly involved, pH, nitrogen to phosphate ratio, photo period, and light. So it can be easily analyzed here that how the H indicates high values, L indicates the low values. So scores are very much encouraging for us to work upon these parameters. Now, the experimental and computational verification of generated decision tree models. What were these? Validation of decision tree models was done by testing them on most recently published data. That is, whatever we tested in the validation of the model, we all gathered the data from the papers of 22 to 23. It should be noted that the tabulated data in the table was not used during any training or testing step of the decision tree construction. That means in one of the manuscripts, we got the comment that please try to test the data for the year 2023 and 22. So we selected that zone, but this data we didn't use to make the model. That means in training and testing, this matrix was not used. So now the collected data set and the prediction by the model. What actually happened here? So NRE means nitrogen removal efficiency, PRE stands for phosphate removal efficiency, biomass. And so now in the collected data set, what we got is here. And prediction by the model was on the right hand side of the table. You can see that the classification accuracy was around 72%, almost 8 points agreed out of the 11 points, and then again the 9 points agreed, classification agree out of the 11 points, so on and so forth. 87% of the accuracy was found in the biomass production. That means, and biomass production is highly dependent upon those parameters which was signified in the graph, and it is 87%. We are getting the model accuracy to generate a model. Usually in the mathematics we describe that more than 95% of the accuracy is desired, but in the experimental modeling it becomes difficult to achieve that value. So 87% looked pretty high to us. So we started working here. So the experimental validation, in this step, the model and rules generated from the decision tree were validated by the real-time experiments. This was the comment which we got from one of the good journals of nature, that the paper is really very good, but then they were desiring some real-time experiments. So one of my MTech students, along with my two PhD students, Dr. Vishal Singh and Manisha Verma, all three of them started working on this concept, and a total of 38 experiments were conducted to verify the biomass production by design experiment from ML. So next I will be showing some tables for the results. How it actually done? It was how it was carried out: strain isolation and identification, followed by the strain cultivation and biomass analysis, experimental designs using ML results, then followed by the error analysis. Error analysis is a critical parameter in the experimental modeling. This is because sometimes the model may be right, but the error values may be very high. Just by you're not having the dots or the goodness of fit in the curve, it doesn't mean that it is an errorful model. That we have tried to describe in some of the research papers, that why such amount of error is coming, and it is all about the disparity in the errors, and the results are only because the different models have been used and the environmental conditions may not be too suitable for the macroalgae to grow upon. That is why we have used the pre-treatment methods also to improve the model efficiency. Now, strain isolation and identification. Two strains were actually isolated. From one was called as VSCM1 and 2. Possibly the students have given their name. One was the chlorella, another was Tasmatismus. They all were taken from the Ravidas Ghat, which is on the bank of the holy river Ganga in Varanasi, and isolation was done in the month of October 21. Then another was collected from the sewage treatment plant, which is situated at Bhagwanpur, Varanasi, and there the complete sewage of total sewage of the all complete city or the villages which is in and around Varanasi get discharged there. What they do, they are doing the primary treatment of the wastewater, followed by the some hard sludge, or what you call as, I'm just not getting the term, what they make. This technology they use. So now we just did some bioinformatic analysis, and the close similarity was found with the Troboxy 5C for the strain 1, it is VSCM1, and VSVM2, there was a close similarity with the class Chloro-5C. The similarity was around 98.54% to 99.32% respectively, and accordingly we got the NCBI registration numbers. So this is all about the phylogenetic tree. This is a well-known concept that we have a bootstrap value, there's the branches, there's a tree, and then the neighbor joining hood method was used to plot these trees. Now the wastewater preparation, how we did. For validation, 3 types of the wastewater were selected: municipal wastewater, sugar industry wastewater or the effluent, and the livestock wastewater. Municipal wastewater was easily available to us. We have several hostels in our premises, and then faculty housing. And livestock wastewater, we have already a dairy unit in the BHU itself, just besides my department, that unit is there. So we collected the dairy effluent from there. Then motivation, and then sugar industry effluent was selected from a nearby industry situated in the Varanasi borders. At Varanasi border it is, in between Varanasi and Jaunpur. The motive behind selecting the sugar industry is because it is very much prominent industry in the South Asian countries. India is also producing a huge amount of sugar out of sugarcane. We also have a good production of sugar canes. That is why we selected the sugar industry. Another point is that since the effluent is from sugar industry, it will have a high content of carbon. So that may help us in solving our purpose. The composition of all the synthetic simulated wastewater are presented in next table. So this is what is the composition is. We just try to make it in our lab also sometimes, because it is not possible. The concentration or the profile of the wastewater which we actually obtained, there's a significant problem in that. Every time we collected the batch of these wastewater, there is a significant difference in their composition. So what we decided, we just standardized the entire process and made a synthetic simulated wastewater in our lab itself, which can mimic the exact condition of those industries or those units. So this is how we made, and we also selected some references out for that. So these are the photobioreactor system, which was actually used. This is the 3D drawing of that. How we maintain the power supply, how we maintain the time. There was electric regulator. We supplied the acid and base to maintain the desired pH values for the macroalgae. Then air compressor system was there. We supplied the CO2 to them continuously for the growth. Then peristaltic pump, pH probe, and thermometers were used to control the flow rate, pH of the entire medium, and the temperature of the entire box. And thermostat, possibly he is missed in the figure. We also use the thermostat to monitor and to tap the temperature at a particular set value, preset value, by using some control devices, which worked upon the Laplace transformation. Then the distilled water supply was there. So this is all made by the software called as AutoCAD. As version type is student type, free license available. So the verification of the results. Sorry. So these are the different results which we obtained. Strain I have tried to show on the extreme left. Then wastewater source, cultivation type, parameters, different type PTM values, reactor type, CO2 content, temperature, illumination intensity, that is the photo period, pH, light intensity, photo periods, the nitrogen to phosphate ratio, class, theoretical class classification, experimental classification, and then experimental class in terms of low or high. So significantly, we got very good results. And naturally, these are where the parameters which are actually shown by the data of 2010 to 2022, that these parameters influence the values of biomass production in any photobioreactor system, especially when you treat these kind of wastewater, municipal, livestock, and dairy. Now, again, the results I've tried to show. There is nothing so unique here. The results were very unique in the sense of what we got as a productivity. And productivity, that too in terms of only 2 strains we utilized, that too isolated by our labs. So the results of 28 experiments resulted in high class level out of 38. So the accuracy is around 74% for the model, compared to the computation accuracy of 84%. Error of 10% is noticed after the experimental verification. This usually happens since we have used the synthetic simulated wastewater. We are not able to give exact natural conditions to the macroalgal cells. We are using a synthetic base and synthetic acid, which is scarcely done by the nature. So that actually hampers the modeling efficiency. Now, around 10% errors seem to be fair enough, as biological data suffers from variations due to environmental conditions. Just to summarize in 2 lines, error can be minimized by constructing more intensive data set and conducting in-depth research on the most critical input parameters. Some other inferences after the experimental verification, which what we obtained is VSVM2 strain achieved higher biomass productivity in all types of wastewater as compared to first strain, VSVM1. And livestock wastewater supported higher biomass productivity than any other category of wastewater. That means dairy wastewater has a strong tendency for the higher productivity than any kind of wastewater. So we did a CAB, that is a cost benefit analysis of the LED applications for the macroalgae, and this data we published in very good journal of critical reviews in biotechnology, published by Vishal Singh. And then input parameters for the electricity cost, we did some calculation work. This was asked by the reviewer to show that what is the efficiency of your reactor in terms of energy, and we just predicted some data. This is all about the benefit analysis of the system. Then 2 basic formula we used to calculate the investment cost and the current and electricity cost. How we calculated this all. So these were the results actually, and the results indicated that here in the study, a total cost, very small amount of energy requirement is required for the propagation of macroalgae. Nothing very big is required here, and most of the research papers, they have not shown this data, and these experiments are actually very expensive in nature. They try to reduce the experimental cost. So just a small analysis we did. Now, the approaches for decreasing the cost of the light sources, because still the cost looks very a little bit higher side. So we just propose some idea of flanging the different kinds of photovoltaic powdered photobioreactors inside the system. This photograph is already taken from a journal of critical reviews in biotechnology. I must acknowledge that, published by one of my students and me in year 2022. And so this we have tried to show here the details of a IGP photobioreactor system, and you can see here that we have used the glass walls. We just proposed it actually, and the different thicknesses and how it can work in the synthetic simulated environmental conditions. So what were the results we actually got is here. That is the some controller mechanism we utilized in this cellular system, this reactor system. So what was the mechanism to lower them the cost was Arduino control microcontrollers. This is one of the best ICT controller till date which has been found to reduce the operational cost of any plant. We use it in different food industries as well. Now we are trying to implement in the wastewater industries. It's completely automated control system having the LDR sensors and Arduino controller sensors for controlling the power supply to the LEDs. And nowadays it has been used in the SCADA system to control the power transmission all across the different dimensions of the industrial sector. So now we have a LDR circuit here. Then a sun was used as a source of energy that controlled and relayed the power circuit. And then we have LED light intensity in the photobioreactor system, which was controlled by the sun energy, totally by the motor and shaft, which was doing some agitation process inside this microbial liquor, where the growth of the LED was taking place. We have shaft process, which is simple CSTR kind of concept. Air pump was continuously giving the air and the CO2 supply for the proper growth of the LED. Then again, the LED support system was used. It was completely made in a box of thermocol using some thermostat to control the overall temperature. That too, we conducted this experiment somewhere around the rainy seasons. So temperature was very much optimum. So there is no input of energy required. In some of my experiments, when there was extreme environmental conditions like 4 degrees centigrade of temperature in the month of December or January, we use some blower concept, use some heaters or convectors to maintain the temperature of the reactor. So now the machine learning presents a sustainable approach for optimization of macroalgae based wastewater treatment. Rules extracted from the present study can be used for designing the future experiments. They can be used at pilot scale, large scale studies without further laboratory experiments. Initial inoculum level and nutrient concentration need to be further optimized, and then the sole application of LEDs for macroalgae cultivation is not feasible. Some of the references which I have used for this presentation. Then we already have the media coverage of the work. Possibly he has not shown, he has also been granted with one patent, Indian patent, on the macroreactor design, and that too, I believe it was a design of a raceway pond system for macroalgae system. So this is all about my research. And thank you for your time and patience.
Thank you so much, sir, for such an insightful and elaborate presentation on wastewater treatment, microalgae wastewater treatment, and focusing on machine learning and the importance of free data sets and emphasizing on experimental design and giving so much technical specification for the continuation of the microalgae. So I'll just go through some questions. So we have a question from Yagi. Were there any calculations involved in identifying diatom predation by zooplankton? Could you please inform how settling velocity or sedimentation rate was utilized?
Sorry, can you repeat the question?
Yes, sir. Were there any calculations involved in identifying diatom predation by zooplankton?
Okay, but we have not worked on that. In
Okay,
case
so-
he's
and,
interested in knowing about the sedimentation of the
Yes, sir.
macroalgae, that system was completely agitated by some mechanical assembly. It was having shaft and everything. So sedimentation of the
Okay.
cells is not possible. Just to break the sedimentation and for proper mixing, for maintaining the uniform concentration of all the nutrients all across the volume of the reactor, including the product itself, we were doing agitation. So why one should expect the sedimentation out of it?
Okay. So we have another question from Ritu. Which tools do you recommend for optimization of the different parameters involved in the cultivation system of microalgae per diameter?
Per?
So it is which tools do you recommend for optimization of the different parameters involved in the cultivation system of microalgae?
So, okay, or diatoms probably. Last phrase is or diatoms. I would like to recommend if you can keep a tab on the CO2 concentration, then photo period, illumination light intensity. And these are three basic, or otherwise it may also be controlled by the type of wastewater and the type of pre-treatment you are giving to the wastewater before carrying out the experiments. What I got in my results, autoclaved washed wastewater gave the best result. And naturally, it is now free from the contamination, so it may be a very good source of um, growth for the macroalgae system.
And then we have another last question by Tree4. He has a request saying that to provide a brief description regarding the slide number 31, the experimental verification, and more specifically the supply of external CO2 to the PBR and purity of CO2.
Sorry, slide number 31. Okay.
Yes, sir.
This is the slide where I've tried to show the experimental setup for the verification of decision tree. This is a kind of reactor system we have made in the lab. What actually is required, please let me know. Possibly I
So
have
he was
also
telling
shown the dimensions.
he wanted some experimental clarification specifically on the supply of external CO2 to the PBR and purity of CO2.
Air compressor or SCU2 supply. Sorry, what technical he wants?
So he was just telling he wanted to know about the experimental verification on the supply of external CO2 to the PBR and the purity of the CO2.
Acha, that means he is more or less bothered about the gas which is being supplied for the
Right.
PBRs. So that actually depends on what supplier is giving to us. Of course, it is not 100% CO2, all of us know. But what cylinders we use for the supply of the oxygen, or sorry, supply of the carbon dioxide, we also utilize the same cylinders for the GC, that is the gas chromatography. So the purity was around 90 to 95%.
Okay, so we have so, I would like to thank you for giving us your precious time. And please stay with us for the session.
Same here. Thank you. So can I leave now?
Yes, sir, it will be fine now. But you can keep attending the session.
Thank you so much for the opportunity, for giving me an opportunity to present my work. Thank you.
Next, my colleague, Mr. Yagik, will introduce the next speaker.
Hello, can you hear me, Dr. Pawan? Hello, I'm Yagik, and can you all hear me?
Yes.
Okay, thank you. Hello,
Can you
I'm
hear me now?
Yagik. Yes, I can hear you now.
Thank you.
I'm Yagik, and as a member of Liquid Trees, I'm pleased to welcome our distinguished speaker, Dr. Pawan Jyother, who will enlighten us with his diverse perspective on our next theme, which is,